Hi, Dr. Rothenberg. Can you set the stage by briefly describing the barrier function, Th2 immunity, and eosinophilic esophagitis?
Yes, the barrier refers to the region of the body that comes in contact with the external environment, particularly at mucosal sites. The barrier is thought to be the first line of defense against insults. Th2 immunity refers to the types of immune responses that typically happen in allergic inflammatory responses and parasitic infections. Eosinophilic esophagitis is a severe tissue-specific allergic disorder of the esophagus associated with eosinophil accumulation. In this particular study, we looked at eosinophilic esophagitis, particularly because it provided an opportunity for unique research experiments because biopsies from human allergic tissue were available through routine endoscopy procedures and hence allowed us to probe human allergic inflammation at a very deep level.
Thank you. Tell us a bit about the approach you took in this study to address the pathogenesis of allergic inflammation and what the results tell us in this regard.
In this study, we started off by examining the biopsies, looking at mRNA profiling through whole genome transcript analysis. We were particularly struck by the finding that there was a downregulation of protease inhibitors, particularly of the family of serine peptidase inhibitors of the casal type, particularly casal type 7, referred to as SPINK7. This particular set of protease inhibitors were expressed at high levels in differentiated human esophageal epithelium. We showed that it was part of the esophageal epithelial differentiation program. Both SPINK5 and 7 were markedly increased during esophageal epithelial cell differentiation. Most notably, SPINK7 as well as SPINK5, but more notably SPINK7, were markedly decreased in patients with allergic inflammation. We then probed the role of SPINK7, particularly by manipulating its expression level by gene silencing as well as gene deletion. We were struck by the finding that deficiency of SPINK7 in esophageal epithelial cells was sufficient to induce a barrier dysfunction, particularly loss of barrier function, as well as transcriptional changes characterized by loss of cellular differentiation. For example, loss of SPINK5 and filigrin. We thus placed SPINK7 silencing or loss of SPINK7 as upstream to loss of SPINK5. We showed that SPINK7 silencing was sufficient to induce the production of large amounts of pro-inflammatory cytokines. Most notably relevant was production of TSLP. TSLP is a protein that is associated with induction of Th2 immunity, particularly because of the polarization of lymphocytes through dendritic cells to produce type 2 or Th2 cells. It's notable that the TSLP gene is located at one of the primary susceptibility loci for the disease eosinophilic esophagitis. We implicated a particular protease itself in the SPINK7 pathway, particularly one called uroplasminogen type activator or UPA. We showed that the UPA receptor was expressed on eosinophils and that activation of UPAR induced eosinophil activation. Furthermore, we proved that this pathway may be pharmacologically targetable by replenishing antiprotease activity using alpha-1 antitrypsin, a prototypic serine protease inhibitor that was readily available and already clinically approved by the FDA for usage in other indications. We showed that treatment with alpha-1 antitrypsin reversed the pathological features associated with SPINK7 silencing. Finally, the relevance of this pathway in vivo was supported by genetic epistasis and variants in the TSLP gene as well as the UPA gene plow.
Thank you, Dr. Rothenberg. The notion of proteasis, anti-proteasis, has precedent in other diseases, notably atopic dermatitis. How did identification of SPINK7 change the way we think about allergic mechanisms?
Yes, proteases and antiproteases have been implicated in a number of the diseases. In particular, very rare disorder called Netherton syndrome is associated with loss of SPINK5 through loss of function mutations in both chromosomes, an autosomal recessive presentation. This is a rare disease, but it is induced, associated with a hyperatopy syndrome, including eosinophilic esophagitis. So our findings in this particular disease showing that SPINK7 is upstream of SPINK5 provides a mechanistic understanding of the linkage between 5 and 7, but also indicates that loss of SPINK7, which is commonly occurring in type 2 responses, particularly of the esophagus, may be a shared mechanism, but one that's more likely to be relevant because it doesn't require rare genetic mutations as occurs in Netherton syndrome.
Thank you, Dr. Rothenberg. What are the future implications of this study in terms of treatment of allergic diseases and EOE specifically?
The implications of this study is that the disease appears to be mediated, at least in part, by an upstream loss of antiproteases. We propose that protease inhibitors are part of a sensing mechanism that detects perturbation in the tissue, and that production of high levels of antiproteases is part of a homeostatic pathway in the epithelium. Loss of the antiproteases is certainly one of the primary events that we propose that leads to a cascade that induces allergic responses. The implications of this is that restoration of antiproteases may be therapeutically beneficial, and we show proof of concept for this, particularly in an in vitro system, that alpha-1 antitrypsin may be an example of a type of antiprotease that should be used for treatment of the disease. We also imply our results that SPINK7 replacement therapy would be another therapy to consider.
Thank you. Dr. Rothenberg, what are some of the unanswered questions that you see as a priority for future research?
A major question that we are currently undertaking is what are the molecular, transcriptional, and epigenetic events associated with loss of SPINK7? How can we understand those, and how can we manipulate those as well so that we can restore the antiprotease activity?
Thank you, Dr. Rothenberg. It's been nice speaking with you today.